Welcome to MSBTalks, a podcast about science for everyone. Our guest today is Dr. Muneesh Tewari, who is currently Professor of Internal Medicine and Professor of Biomedical Engineering within the University of Michigan Medical School and College of Engineering. His lab currently studies liquid biopsy approaches for early cancer detection, the role of innate immunity in CAR T-cell therapy toxicities, and wearable sensors and machine learning for disease prediction. This episode, we talk about Dr. Tewari's journey through scientific background, research interests, and thoughts on the future of cancer detection and immunotherapy. This is MSB Talks. We're glad to have you. And now for our conversation with Dr. Tewari.
All right, thank you, Dr. Tewari, for having me. It's a pleasure to be in your office. Before we kind of delve into the technical aspects of this interview, I was just wondering a little bit about yourself, how you got to where you are today, and that kind of thing.
Oh, sure. Let's see. So I'm a faculty member here now at the University of Michigan Medical School. I started out, I grew up mostly in Detroit.
Yeah.
And then just the educational history. I was at Case Western as an undergrad doing biochemistry there. And then actually came here to the University of Michigan in the MD PhD program and also did medical residency here in internal medicine. And then I have been to both coasts. I went to Boston to Dana-Farber Cancer Institute and Harvard Medical School to do oncology, medical oncology. It's a clinical fellowship and then a postdoc there in systems biology. And that's actually when I got introduced to synthetic biology as well. And then I went to Fred Hutchinson Cancer Research Center in Seattle. I was faculty at University of Washington and at the Fred Hutch for 9 years, and then came back here to University of Michigan about 12 years ago. My primary department is internal medicine with hematology oncology in that division, but I actually do have a joint appointment in BME here, biomedical engineering, and I also have a role as co-directing the Center for Computational Medicine and Bioinformatics, which is in the Department of Computational Medicine and Bioinformatics. So that's kind of the overall, like a biography of how I got here.
So, why specifically did you choose to pursue like an MSTP program as opposed to just an MD or just a PhD?
Yeah, at that time, I think that was, of course, a big question, a big decision. I think I knew at that stage I had done undergraduate research. I'd even been fortunate in high school to work at a place called the Michigan Cancer Foundation in Detroit. And I think I pretty much knew that I had the research bug and it's something I wanted to do.
Yeah.
And at that time, it was an opportunity to really go deeper into research and take that time, because it's a particular time of my life too, right? At that time where I could take that time to go deeper into the research, into the foundations, even into some of the philosophy of science. And so I think that's why I chose that path. I thought it would just give me, the biggest background. And a deep background. That said, that's not the only way to become a physician scientist and to do this kind of work and to do it really well.
I see. Yeah, so obviously a lot of your research centers around cancer biology. Is there any topic or specific topics that kind of interest you most or that you're most excited to look into?
Yeah, I think, for me personally in our research, it's really evolved. I started out doing very fundamental molecular networks involved in cancer, and even using model organisms, using cancer systems. We studied microRNAs for a while.
Yeah.
I would say if you ask me today where our research has evolved to and where my research interests have evolved to are the relationship between the mind, the brain, and cancer outcomes. I feel like this relationship of how the nervous system interacts with cancer cells, interacts with the immune system, and how it really ultimately affects clinical outcomes. That's something that's really been not explored anywhere near the depths that I think it should be. Because I think there's a lot to be learned and a lot to be applied from there. In the last 5 or 10 years, there's been a lot more interest in this. There's this whole area called cancer neuroscience that people are exploring. Yeah.
So, in regard to the nervous system, is this specific to cancers in the nervous system or just cancers anywhere in the body?
No, cancer anywhere in the body. And the basic, I guess, idea is just that virtually all tissues of the body get not just blood flow, right? Because you get arteries and veins, but typically there's also nervous system connection,
Yes.
because there's these neurovascular bundles that are actually going through the body to all different tissues, and it's just that I think the idea is basically that those nervous system signals actually matter, and that they're affecting not only the tumor cells but their microenvironment, even their even the local and peripheral immune system.
I see.
Yeah.
Yeah, I'm glad you brought up the microenvironment because it kind of gives me a chance
Yes.
to introduce our project. So as we discussed a little bit earlier, our project essentially involves taking a non-model bacterium, AMB1, it's a magnetotactic bacterium, taking that, enabling or giving it essentially a dual plasmid system, one of which enables it to localize to ovarian cancer cells by expressing a ligand for folate receptor alpha, and the second of which essentially enables it to kill those tumor cells with cytolysin. Given this kind of broader approach and its similarities to precision medicine strategies such as CAR-T, etc., is there any kind of insight that you would offer in this regard, given your experiences?
Well, I think what I really like about what you're doing is that localization part, of course, because I think that's one of the challenging things with a lot of therapies, and how to avoid off-target effects.
That's true.
So as I understand it, you're basically using the bacteria as a therapeutic agent,
Yeah.
right? With the cytolysin
Cytolysin,
A, I
yeah.
think. Yeah, that's a toxic protein to deliver to the tumor cells. But then you can turn it on or off, I guess, using the alternating magnetic field.
That's right.
Is that right? Yeah. Yeah. So I think what's really appealing is the fact that you can actually make it more precise, both in time and also in space, right? Through these methods. I think the important thing, of course, will be to figure out what's the right use case, and this is maybe down the line. I mean, the first step is proof of concept.
That's true.
But also, where can you line up being able to localize the tumor, deliver the alternating magnetic fields, and then also empirically, getting a high localization of the bacteria to the tumor. Those are some of the things that will be in my mind.
Gotcha. So I guess delving more into the topic of CAR-T, a big kind of focus there is exactly what you said, just ligand specificity, right? When we talk about folate receptor alpha, we know that it's overexpressed in, I believe, like 70 to 90% of ovarian cancers, right? But there's obviously that 10% left over, right? And there's always this fear that, folate receptor alpha is not exclusive to ovarian cancer cells. It's expressed in a number of different cell types. If we're only talking about just expressing the ligand and kind of tossing magnetotaxis and airtaxis aside for now, have you encountered some strategies to kind of make that interaction more specific?
I think that's in general been challenging, I think, in the field as far as I've seen. I think one of the reasons CAR T-cell therapy works really well for example, B-cell lymphomas or B-cell ALL, a couple of indications where it's been approved, is that it, for example, targets CD19, which is a protein that is expressed on cells in the B-cell lineage, but not really much anywhere else.
I see.
And so what happens there is that all the lymphoma or leukemia cells that express the CD19 can get wiped out potentially with the CAR T cells, but also all the, basically the normal B cells, or a lot of the normal B cells, are also being wiped out. But because the body can tolerate that, in general, I mean, it does lead to immune suppression, but in general, it's not, acutely lethal, right? Or that has succeeded. Now, if that CD19 protein was also present on lots of other tissues, I don't think it would have succeeded in the same way. And so I think where the successes are, one of the key parts of success of CAR T-cell therapy that we're seeing is a very specific, receptor. So I do think without the localization of your, of your alternating magnetic fields and being able to target it that way, I think it would be hard. Just trying to go for specificity just with that cell surface protein receptor. It will work for certain cancers.
I see.
Like I said, CD19 in this case. There's other examples of very targeted, like very specific protein expressions, like PSMA is one in prostate cancer.
I see. So essentially with CAR T and this kind of other approach, do you think like the best use of our time is to just find cancers that to which it's better suited as opposed to like optimizing it for one specific cancer?
You mean your approach with the folate receptor is what you're talking about?
I mean,
or?
like, generally speaking, right? If you have, like, certain cancers that are more specific in, like, the receptor that they
Yes.
overexpress, is it better to just dedicate more of your time, like, optimizing the approach to those cancers?
Yeah.
Or just, like, looking at a single cancer and trying to optimize your system
Yeah.
for that?
You know, it kind of depends on what your objective is.
Yeah.
So I think, in general, from what I've seen, right, it's in the end, it's like a mix of both
I see.
of the overall strategy. But if I look at how CAR T cell therapies have been developed, in my perception, they kind of went for where is the right protein target cell surface where they're likely to not get a lot of toxicity and side effects, like I was saying, because it's only expressed really on B cell lineage,
Yeah.
but also is highly expressed on tumor cells. So I think that's kind of the approach that, in general, probably the drug development takes in this kind of thing, is first start with that because it's kind of the lowest hanging fruit.
I see.
But and then sometimes and then try to expand. So that's kind of what's happening in the CAR T field right now. People started with those so-called liquid tumors, and now they're trying to get into more broadly into solid tumors, which have different challenges that they've been trying to solve. So I think it depends on what your objective is. On the other hand, if your interest is only in one particular cancer and that's what you care about,
Then you should
you should go that way and try to solve that problem. But to me, they always go hand in hand because the first way is also informing now the second way, right?
Yeah.
Like, so what you learn from trying to go for the low-hanging fruit will help you, to then go for, let's say, a very cancer-specific thing.
Yeah. Actually, something you brought up about like liquid versus solid tumors was something I was going to ask about.
Yeah.
We know that CAR T is like characteristically like much more effective at treating, cancers in a non-solid state than it is for solid tumors.
Yeah.
Out of curiosity, do you know like why exactly that is?
Yeah, I mean, I know it at some levels, and I think there's also a lot of unknowns of why that really is. Some of the things I think people have seen is I mean, even just the physical matrix of a classical solid tumor, right?
Yeah.
Just to penetrate and so forth. There are those things that can be desmoplastic reactions of a lot of stromal cells, and so that's one of the things that people have talked about. There's also perhaps the tumor microenvironment there is more complex and more immunosuppressive. I think that's another challenge that people have had. Also finding very, very specific targets, protein targets that are not, expressed in a lot of other places. Those are, I think, some of the things. At the same time, I think I'm sure there are some variables that we probably don't even fully realize right now. That's my perception of it.
Yeah. Well, I guess we'll kind of shift away from CAR T now. Another thing that I talked to you about earlier were antibody-drug conjugates, or ADCs. They do have a few parallels with our project, right?
Mm-hmm.
For one, there are already ADCs for ovarian cancer that target folate receptor alpha, right? And they're bound by a peptide linker to some, chemotherapeutic payload. Our project varies in the sense that while we do still target folate receptor alpha, our payload isn't even expressed essentially until docking at the tumor site is confirmed. Does that have any advantages or possibly disadvantages? And if so, essentially, what do you think of those?
Yeah. I think one of the biggest advantages is really the likelihood that you're going to have less toxicities. I think that's one of the challenges with the ADCs. Even with folate receptor, like it's not, as far as I know, it's not 100% specific, right? Only to ovarian cancer cells.
Yeah.
And so with ADCs, you get off-target effects. You get, a lot of the toxicity is related to that. But with your system, because you can locate it in space, ADC, right? The drug is going all the way throughout the body. But in your system, I think because you can localize the activity in space, I think that could be a big benefit. The other thing I think that I think is just underexplored in oncology in general is time. Because because normally those drugs are just given on a kind of a standard schedule.
Yeah.
But the timing of that hasn't really, usually is not studied very much. In terms of clinical trials, and so I always wonder, trying to maintain the steady state or just trying to give these drugs at just very fixed intervals, would it be different? Is there an adaptation, right, that happens of the tumor cells over time, or even a selection?
Mm-hmm.
And could you actually potentially get around that by the fact that you could control the timing, and you wouldn't necessarily? So if you wanted, you wouldn't have to just give it continuously, but you could give it episodically, and you could experiment
stagger
with that.
the doses.
Yeah, stagger the doses and experiment with that, because you have control over that a lot more easily. So those are some of the thoughts. I mean, I think that second part is quite speculative, but I think the first point of being able to localize where you're releasing it to reduce toxicity, at off-target tissues and organs, that seems pretty solid to me, at least conceptually.
Yeah, and then another topic that we had been discussing earlier was this idea of imaging, right? Obviously, if you're using like a cell culture, it's pretty easy to see where your cells are if you're using like a fluorescent marker, which we are. But obviously, in an in vivo system, whether that's in a animal study or like in clinical trials, it's a little bit more difficult to kind of detect where your bacteria are, essentially, right? So what advice would you give kind of in this regard?
Well, I mean, as far as I can tell, you're an early stage, right?
Yeah.
So, I mean, the advice I would give is think proof of concept, but also think down the line. So it's kind of like thinking about those both things together.
Mm-hmm.
Because in the early stage, proof of concept is most important. But as you design the path of how to take this work right to the clinic, having a vision for where it's going and what the constraints will be there, I think would be valuable.
Mm-hmm.
So for an ovarian cancer in particular, a lot of the small micrometastases or peritoneal metastases, for example, are sometimes not easy to image, even on CT scans. But on the other hand, there are big masses, right, that can be imaged even by ultrasound.
Yeah.
And so I guess in the developmental path, there's just so many things you have to kind of line up. But thinking about what would the clinical application be, could it be perhaps like debulking or reducing the size in advanced disease, in recurrent disease, and just sort of, I guess, think about what the use case will be. And of course, the best advice for that is to actually talk to ovarian cancer clinicians, oncologists who are treating ovarian cancer patients a lot, and to get that input.
Yeah. And do you have like broader experience with like translational medicine, like, and knowledge in that regard?
Yes, that's like I think a lot of what we do.
Okay.
Yeah.
So I guess that kind of follows into this next part, but taking like a proof of concept or like a lab discovery and turning that into like an IND, right, an investigational new drug. What hurdles would you like canonically or like characteristically expect in there?
I mean, typically, it kind of depends like on what the safety profile. That's one of the first things is. Of whatever you're taking forward. So, for example, with one of my collaborators in our larger team, we're taking a drug that already is FDA approved and repurposing it for another purpose. So, in that case, some of the safety hurdles are a little less. Because the drug's already been in humans and so forth, right? So, I think one of the big initial things really for the IND and for the FDA is safety.
Yeah.
So, the biggest thing is just thinking about that and then working with the FDA to find out what kinds of preclinical or, animal model or other types of data, that experiments that you need to do that the FDA would consider, giving sufficient proof of safety to be able to go into humans. That's really, I think, the big one. I mean, there are lots of other things we have to think about in terms of manufacturing, stability of the agent.
That's true.
Lots of other operational things, but safety is the big one, especially if it's completely new type of treatment.
Yeah. So effectively, when you're trying to get a regulatory approval, safety kind of takes precedence over efficacy, or are they kind of balanced?
In my view, like safety does take precedent because the efficacy will be tested in the clinical trial. And usually, and oftentimes, you'll have an initial clinical trial, it's still a clinical trial, but it's not of efficacy, it's really of safety.
So they really won't let you progress to further trials until you've proven safety.
Yeah, exactly. That's the first step. Efficacy comes later.
Yeah.
Yeah.
I can imagine the hurdles to proving safety are pretty high. Right?
In some ways not. It's very use case or very case specific, right? So if you're using something that has already been, maybe the components have already been used in humans before, then the hurdles might be a little bit lower because there's already information there. But in terms of actually doing the studies, I don't think it's that there's huge hurdles. It's just that maybe the amount of experiments you need to do and how many things you need to think about will be more, if it's a completely brand new treatment.
Yeah.
So I guess how you look at it, I guess, whether it's a hurdle or there are more steps, maybe is another way to put it, yeah.
And do you know what kinds of specific experiments are kind of needed for that?
Well, again, it kind of depends
on
case specific, but, if these, typical ones, oftentimes, if they're potentially toxic drugs, right? Oftentimes they're experiments in animal models to see what are the side effects, what other organs are affected.
Yeah.
In this case, like if it's these bacteria, I imagine there'll be questions about, what is the pathogenic potential of the bacteria? Have they been already put in humans before? At what doses? Various things like that. Could bacteriophages infect
That's true.
the bacteria? Could you get problems that way? What if the bacteria go rogue? How are you going to shut them down? Do you have a
essentially
mechanism?
You got to explore all possibilities, right? Murphy's Law.
Yeah, exactly.
Whatever can go
Whatever
wrong.
could go wrong.
Could
Think about all those things. And maybe you further engineer these bacteria if that turns out to be a concern, to have kind of a kill switch, people call it, or whatever, right? Like where you could give some other drug that is then becomes very toxic only to the bacteria because you put a gene in there, for example, that metabolizes a prodrug into a drug or an antibiotic.
Yeah.
I think something like that.
Your mention of a kill switch, we don't have like a specific like chemical tag that we would kind of target in this.
Yes.
But what we would use is, again, that AMF device.
Yeah.
I mentioned that it can reach critical temperatures for heat shock, right?
Yes.
But of course, if you intensify the AMF, right, increase the speed at which those dipoles are flipping, you can increase the temperature of the system, right? And perhaps you can reach a temperature at which it's not permissive for those bacteria to survive.
Right.
And in turn, that could act as a kill switch. Does that seem viable in essence? Or is that kind of less specific than a chemical tag?
Potentially, but I guess, that'd be my concern is like, would you need to do that then throughout the whole body? So would you need a whole body alternating, magnetic field, and then would you induce toxicity to other places in the body?
That's true.
Because there are still hypoxic places in the body. It's not only tumors.
Yeah.
Right. So, are you going to, like, in the intestine, are they going to end up there and create this toxin? I don't know. I mean, those will be the questions that come up. Yeah.
Yeah. I think it's always like an interesting topic, especially when it comes to this idea of like bacterial therapeutics, quote unquote, right? Because it's a relatively little explored field. Have you heard of any other bacterial therapeutics or?
Yes. Well, I've heard of, I don't know how far they are into the clinic, so I can't say that I know much about that. I haven't followed that, but I know that this was probably around 20 years ago. There was interest in Bert Vogelstein's lab from Johns Hopkins and some other, I think, were really interested because what they found was, I think, injecting bacteria, and I can't remember exactly which species they were, but basically they were trying to use them but they were also anaerobic bacteria with a kind of a similar concept, at least in part, where the bacteria would go to the hypoxic tumors regions, and I can't remember how they were actually inducing. Actually, I don't think they had a very like a synthetically engineered toxin or anything like that, but I think the idea was that they were using that to then cause tumor cell death, and they saw some promising results in animal models. And I believe when they studied it further, it actually was also related to the immune system. and there was some kind of immune reaction perhaps. So I haven't kept up on it. I'm just giving you a very superficial thing of my memory, but you could look it up. I think one of their papers was in the PNAS, the Proceedings of National Academy of Sciences. But I think that just brings up another topic, which is, what's the how is the human immune system going to interact with these bacteria? And I think that's a question that will, come up also in the
certainly
safety and all of those effects.
Yeah, I think we've read a decent amount of literature proofs demonstrating that AMB1 is like non-pathogenic, that it doesn't induce like a septic or inflammatory response.
Yes, yeah.
But obviously there is that inherent risk when you're introducing like foreign bodies into the human body, right? Our understanding is that AMB1 essentially, once it does its job, kind of passes through the digestive tract and then leaves the body. But do you know of like any other kind of ways in which we could evaluate the immune response?
Well, I mean, I think some things would be just to see are there antibodies made against the bacteria, when you inject them?
I see.
Are there lymphocyte reactions against the bacteria? Those would be a couple of, I think, the big ones. I guess the other thing is, are you thinking about this just orally? Because then they would only stay in the gut, or is this kind of like a, IV injection, which I assume?
I think it is IV.
Yeah.
Obviously, that's really downstream.
Yes.
But yeah, ideally it would be intravenous, and then you could use like an external magnetic field to also kind of guide those bacteria to where they need to be. But yeah, what would be like the primary difference in between like an oral administration and like IV administration?
Well, I think the big difference would be that if you gave it orally, unless somebody's gut is not healthy, in general, those bacteria would not become systemic. They wouldn't get in the bloodstream, or at least not at very high concentrations. And so you might have a lot less delivery to ovarian tumors.
That's true.
Whereas if you give it IV, they can go directly through the bloodstream to the tumor.
So in general, like with cancer drugs, in what cases would you administer it orally versus like IV?
Yeah, so since cancer drugs, a lot of them are small molecules, so those can get absorbed through the intestine. And so some cancer drugs can be given orally. It's just that if you're giving a bacteria, it's so big, and there are already mechanisms of the gut-intestinal barrier to try to keep bacteria that are in the intestine out of the bloodstream, right? There are evolved mechanisms for that. Similarly, like, antibodies and things like, there's proteases, things get chewed up. So it's usually the small molecules that don't get degraded in the digestive tract and then can be taken up by the intestine.
Yeah. Like, I'm not obviously like an expert in oncology at all, but it kind of appears to me that like IV seems like superior to oral administration. Is that universally the case?
Well, I mean, I wouldn't say it's universal, because sometimes the problem is in the gut.
Yeah, of course.
And in which case, IV will be worse.
That's
It would
true.
be better to give antibiotics.
That's a good point.
But aside from that, I mean, in general, if you have something that you want to get to tumor sites in multiple places, IV is generally going to be better.
I see.
Yeah.
Cool. Well, I think that was a great delve into like the technical aspects of our project. Before I do leave, though, I would like to ask you about some advice that you might have for any budding MDs or PhDs or anyone interested in your kind of research or your work.
I mean, you mean like going down this path of medical research? Is that what you mean, or
anything,
or
anything related to your work? Yeah.
I guess I think it's kind of the general advice, but I think it's pretty foundational. I think if you want to do this kind of work, and you want to do medical research, and you want to, come up with new treatments or new discoveries and all of this, like it really has to be aligned with your personal values. And so first, I mean, it's the classic thing, right? Know thyself, but it's really, seeing if this is something that you have a passion for, you keep thinking about, you really want to, get into, or at least identifying what that is for yourself, right? What is it that really, you don't really have to force yourself to do it, and so I think knowing that is really important. It's really foundational because this kind of work needs a lot of creativity. It needs a lot of persistence. It needs a lot of interdisciplinary, working with people, and so it really needs a lot of energy.
Yeah.
And so the things that are really aligned with, that you kind of have, that you have a passion for, I think, is where you're going to find that energy. So
I see.
So I think that's like a foundational thing. And I think the other thing is, of course, to like become as knowledgeable as you can on the technical aspects of it and try to go deep.
Yeah.
And see both why certain things have succeeded, but also why a lot of things have failed, and try to understand the why behind that. And then in going deep, like ask questions, even if they seem silly, to really get a mental model of how you think things are working and how the whole system is working. Because to me, that's what science is we have a mental model, like we're trying to come up with a model for how everything is working, knowing that it's incomplete, knowing that it's going to change, but at least as a way of making steps forward. And I think the last thing is, if you want to do this kind of work, keep working on, and I'm constantly working on my communication skills.
Yeah.
And not just speaking, but also listening and the whole thing of because I think it's a fundamental thing of being able to work with people from diverse backgrounds and areas and expertise. Those are my thoughts.
Cool. Well, yeah, no, it was great getting to talk to you. I really appreciate you giving us the time to kind of ask questions and such.
My pleasure.
Yeah. All right, cool.
All right.
That concludes this episode of MSB Talks, where we delve into the science behind our research into cancer, therapies, in a hope for a better solution. We're committed to sharing evidence-based insights and the voices of those advancing our understanding of cancer. And to stay up to date with future episodes, subscribe to our YouTube channel. If you have feedback, questions, or personal stories you'd like to share, we welcome you to reach out, as you can find us on our Instagram at @michigan.msbt. Speaking to Dr. Tewari was Arnav Sharma, a member of MSBT, and I'm your host, Daniel Yang, and I thank you for joining us today.